I remember this was one of the first conferences going to as a young researcher, and so it's a pleasure to be back here and an honor to be giving a keynote this evening. It's 5 p.m., but I had a double espresso, so I should be okay to make it through. All of this is tweetable if you'd like. I'm on Twitter at Phillippy, and we'll jump right into it. So this talk is mostly motivated by the new sequencing technologies that are emerging. And in my research career, it's always been a continual process of adapting old tools and methods to the new sequencing technologies that were coming out. First, the Sanger to 454 transition, and then to Illumina, PacBio, and most recently, we're working with the Oxford Nanopore device. So I'm sure most of you have seen this after being at this conference for the last couple of days, but I'll just play a short video to kind of motivate on the algorithmic side why we're so fascinated by this device. First, as you know, it's no bigger than the size of this remote. You can put the DNA on it after a short library prep of maybe 5 to 10 minutes, and you get single strands of DNA going down through the nanopore and being read off as these squiggles, which are readouts of the current as the ion flow is impeded by the bases that are in the pore. If you visualize that then, each of these reads popping off in real time, and visualize it as a coverage plot. This is a bacillus genome, and I'm going to show the genome coverage piling up over time. And so this is in a lapse of maybe 40 minutes of sequencing time. And it kind of illustrates the real-time properties of this device, that you put on your library and instantly this is playing back as a video of elapsed sequencing piling up across the genome. And so after maybe 10 minutes or so, you have 5x coverage. And this was from the device 2 years ago. You get even more throughput these days. Accuracy of these reads currently is about 90%. You can get 10 kb plus molecule lengths and maybe 5 to 10 gigabases per 48-hour run. And so from somebody that likes to develop tools and algorithms, it's just an awesome device to think about playing with because it's an entirely new data type. The reads are long, they have a pretty high error compared to what we're used to, and they're coming at you in real time. So a great problem for methods development. One of the very unique capabilities of this device was illustrated by Nick Lohman's group, in particular Josh Quick, pictured up here, was the development of this ultra-long read protocol, which they demonstrated most recently in the spring. And so Nick has blogged about this. He might have talked about it in his boss talk earlier, but I'll cover it just again to say. They did a human genome prep, put it on one of these MinIONs, and achieved 100 kb read N50 size, right? So this means that half of the data is in reads of length 100 kb or larger, as you can see down here from these distributions. This is the ligation nanopore prep, the ultra prep, and this is the ultra long prep. So log scale here, this is 100 kb, and you can see that half of the mass of the distribution is bigger than 100 kb, with sizes up to approaching a megabase. So a single read of DNA, a megabase long, coming off of the instrument, and this enables great new applications. Just to give you an idea of the scale of this, here's the size of the pore measured in angstroms. So the ONT-R9 pore is actually a re-engineered E. coli membrane protein. So it's really fun to think about an E. coli protein sequencing the E. coli genome so many times over. And the scales of some of this are quite amazing. So if you imagine a 1 megabase strand of DNA going through the pore and we scale it up where the pore is the size of my fist, we're talking about a rope of DNA that's over 3 kilometers long. And so when I look for analogies for these sizes, I always like to just type 3 kilometers into Wolfram Alpha and see what comes up, and this is a good one. So 3 kilometers is the height of a stack of Lego bricks, such that if you add one more to the top, the bottom one collapses under the weight of all of the other Lego bricks. That's your interesting fact for the day. It's also like 8 Empire State Buildings tall. So it's a kind of mind-boggling act of physics that such a long rope can be threaded down through this pore and you can actually read off bases. So what's the problem? Like the Lego at the bottom of that stack, all of our tool chain breaks when we deal with this type of technology because it's an entirely new characteristic. You've got 10 kb plus reads, you have 10% error rate. All of the tool chains that we're used to using break. And the one that was most obvious to us when we started looking at these ultra-long reads is that the BAM format itself breaks because it's based on cigar operations and it's stored in a short int, so they can only handle 65K cigar operations. So if you feed a 1 megabase read in, you align it, you get more than 65K cigar operations, it breaks the BAM format. And so we go through this like every 5 years in bioinformatics, right? So we have to redo that. And it also changes the way that we develop tools, because now instead of looking at the mapping problem of short Illumina reads against whole genomes, we're talking about essentially getting near whole genomes out of the machine if you're talking about viruses or bacterial genomes. And so we really have to think about complete genomes assembled de novo kind of being the norm of analysis again. And just to point out that these tool chains take years to build, if we think back to the last technology revolution when Illumina came on the market, 2006, kind of the introduction of Solexa Illumina technology, and it was 4 years before we had assemblers that were capable of reconstructing vertebrate genomes from that data type, even though the contigs were still very short. And 3-4 years until the popular tools like Bowtie and BWA came out and became the standard. And so we're at kind of the midway point with this Nanopore technology that's been out for 2 years yet, but it's going to be another 2 years or so until the tool chains really get built up that it becomes useful in everyday biology. So since this is a keynote and I have an hour to blow, I'll wax poetic about some of the things that came before me. And just to point out that this isn't entirely a new data type. When we were doing Sanger analysis in the late 90s and the early 2000s, these are hundreds of base pairs long and 1.5% error. It's a little more noisy than Illumina. And this genome assembly problem has been around for almost 40 years if you go back into the literature. In the May issue of Genome Research, I edited a collection of papers on de novo assembly, and part of my editorial from that pulls this cool quote from Roger Stadden in 1979, which is the origin of the assembly problem. And if it reads off, if you just ignore the fact that gel sequencing doesn't exist anymore, you could imagine this in a modern paper where they talk about modern fast sequencing techniques, obsolete restriction maps, and if the 5' end of one read overlaps the 3' end of another read, it's said to overlap. If that overlap is of sufficient length to distinguish it from a repeat, then you can think of it as contiguous, and that's how you get your contigs. And so you see the origin of all of the words that we use now, like contigs and overlaps and reads, from this paragraph from about 40 years ago. Fast forwarding a little bit to 2001 and the reconstruction of a human genome de novo from shotgun data as pioneered by Gene Myers and Craig Venter at Celera Genomics. And I found this old vintage photo of Gene from 2001 actually describing genome assembly on C-SPAN. So those not from the States, it's like an ultra dry, recording of what's going on in Congress during the day. And so Gene magically appeared in 2001 describing the assembly problem, and his hand is up because he's actually pantomiming what paired end read sequencing is to the C-SPAN audience. That you read from one end, you read from the other end, we use that pair information to reconstruct whole genomes. And this pioneering effort really did foreshadow the future that whole genome shotgun assembly really is the way that all of these projects are done now. We don't do back cloning and back sequencing anymore. It's all from the shotgun method. And a lot of that came from the work of Gene. That was right around the time I started working at Tiger Genomics, and we were doing a lot of microbial genome assembly and alignment. And in that position, I was really influenced heavily by these three researchers. The one on the bottom left is Art Delcher, who was the original developer of the Mummer and the Glimmer toolkits for aligning and gene finding in the early microbial genomes. Steve Salzberg, who was leading the bioinformatics team at Tiger at the time. And Granger Sutton, who moved to Celera and did a lot of the fundamental work on the Celera assembler. And so my career was really shaped by these researchers and these two tools in particular, the Celera assembler for alignment and the Mummer sequence aligner for whole genome alignment. And so the rest of my talk will be how we tie those old methods into this new technologies to do a lot of the same tasks that we've been doing for 15 years. Another concept that kind of recurs in my research lately is this concept of the minimizer and its use in genome assembly. And so this was first developed by our friends at University of Maryland, Mike Roberts and Jim York, who were in the physics department there, and they started working on the genome assembly problem, even though Jim had a storied career in physics and has won the Japan Prize and is known for coining the term chaos theory. He decided he wanted to quit all of that and do genome assembly for whatever reason. And when they started rethinking the problem, they came up with this neat idea of the minimizer to speed up the overlapping problem. And it's a very simple idea in practice. It turns out to be very useful. If you take all of your k-mers and you define some ordering on them, in the minimizer paper it's just using a lexicographical ordering, and you read off your sequence, decompose it into all of the k-mers, so I've decomposed it into 5-mers here, and you just remember the smallest 5-mer in terms of its ordering, so here AGTAT. That's the minimizer for the sequence. So if you define a window size, you scan across your genome and collect all of these minimizers. they become useful because if you have two sequences that are similar in their whole sequence, they're very likely to share a minimizer. And so Mike and Jim developed this technique and built an overlapping tool that was used to accelerate some of the Celera assembler at the time. And this was back in 2004. It turns out that Mike and Jim were really onto something. In fact, something that had been discovered prior, back in 1997, in a more general form in the term of minhash. And so Andre Broder was at Alta Vista at the time and tasked with the problem of identifying web pages that were similar so that they could remove duplicates from the web search. And so he came up with this hashing approach where you define a random permutation, which is usually approximated by a hash function, that takes all of your k-mers, just turns them into numbers. And for a document that you read or a sequence that you read, you remember some number of the smallest k-mers that you saw. So if you think about this a little bit, if you remember only a single smallest k-mer, this is really equivalent to the minimizer. But the Broder paper is helpful because it lets you think about this in a probabilistic setting. That if you take a sequence A and you take a sequence B and you hash them with the same hash function, the probability that they share the same minimum value out of that process is equal to the Jacquard index. And the Jacquard index is very simply the intersection of two sets divided by their union. So if A and B are our sequences and they share a minimizer, I have some expectation that they're similar because the probability that they shared that minimizer is defined by the number of k-mers that they share. Broder's formulation was much more general though because he was looking at collections of more than a single minimizer. And so here if you let A be the minimum value out of a hash of the union of A and B, the probability that that minimum value exists in the intersection of your two sketches is also equal to the Jacquard. So if you repeat this process for more than one hash value, you start to get an approximation of their Jacquard value. Because each sampling that you do, think of it as throwing a random dart at that picture, either they share it or they don't, and you repeat that random sampling over and over again, the more samples you collect, the better your estimate of the Jacquard value. And so this turns out to be a very powerful technique that informs a lot of the methods that we're working on today. So I'll show a quick visualization in case you didn't follow that. If we have A and B decomposed in the k-mers, we run it through a hash function that just simply turns them into bags of numbers. So here we have A and B. We want to approximate the Jacquard, which is the intersection of that divided by the union, just in terms of their cardinality. And the sketches, as we run those hash values through the analysis, you just keep the 5, in this example, smallest values that you saw. So if you run it through the hash and it's not smaller than the 5 you're currently remembering, you just simply throw it away. And you only remember a new value if it's smaller than the current values in the hash. You can do this very rapidly, and you collect a sketch, as it's called, for sequence A and sequence B. And the real beauty of this is because you're remembering the 5 smallest from A and you're remembering the 5 smallest from B, you're guaranteed that the 5 smallest values between the union of A and B will be in that set. So if you take your sketch of A and you take your sketch of B and you merge them together and remember the 5 smallest there, you've accumulated a sketch of the union of two objects before you even knew that you needed it. So you just started with a sketch of A, you started with a sketch of B, you could have sketches of C, D, E, F, and G. You take any two of them together and you run it through this process and you can compute the sketch of the union of those, and with that you can compute the Jacquard. And so it turns into a great dimensionality reduction technique because you can take your large documents, your large sequences, run it through this process at the beginning, and then when you want to compare them and figure out their similarity, you're only dealing with a small number of values instead of some gigantic set. In the biological sense, you can use this for a lot of fun things because now you can scale up to enormous quantities. And so one of the first applications that we did was to take all of the genomes that were in RefSeq and cluster them by similarity. So we took all of the genomes, sketched them into, I think at the time, maybe 500 values per genome, so very small per genome. Did all pairs comparisons, which was billions of pairwise comparisons, but you can do it so quickly because it's just one of these linear merges. And here I'm showing the result of the microbial components where each of the colors is a species label, but the clusters were developed de novo by this distance technique. And all of the major groups like E. coli and so on cluster together very well with one another, but don't cluster with things that they're dissimilar. So it's one of these big data approaches for dealing with this. So Brian Ondov in the top right and Sergey Korin are here at ISMB as well, and they have posters at today's and tomorrow's sessions if you'd like to hear more about some of this genome clustering work. But now I'd like to get into kind of the bread and butter of what this talk will be about, and it's using these long reads for getting very high quality, very continuous genome assemblies. So in the spirit of digging up old photos from people, I also dug up a photo of Mike Schatz from 10 years ago when he was still in his ponytail era. And I borrow this slide from him because it's a neat way to visualize building a genome assembly graph. And so if you imagine your DNA sequences as just shards of text, the overlap layout consensus style assembly strategy looks for fragments of text that share a significant number of words. You paste them together and draw an edge between those two sequencing reads if they share a certain amount of words, and you end up with a graph like this. And anywhere there's a single path between a set of fragments, you know you have a unique reconstruction, and anywhere where there's a branching path at the end of one of these fragments, you've encountered a repeat, and you can now either go to a distant or a near part of the genome based on which repeat copy you're going to go into next. If you collapse down those unique paths, you end up with your genomic contigs. So it's very easy to visualize in this way. And with sequencing reads, it occurs very similarly. With the observation, if you have very short fragments, this is a hard problem. So this is the assembly problem for masochists. If you like Illumina data and you like to write Illumina assemblers, you're left with a huge pile of very short pieces, and it's a very hard problem. If you're like me, you like easy problems, and I like to put Dora puzzles together with my kids. And for the same reasons this puzzle is easy to put together, the same reason it is to assemble large genome sequence reads, because each of the puzzles is unique because it's capturing more context of the overall sequence. Where you're dealing with short things, it's hard to tell where the fragments originated from. To give a real example, I like to show this visualization of an actual assembly graph for E. coli K12. If you had perfect coverage and no errors of 50 base Illumina reads, this is the best that you can do. Based on the repeats that are in the E. coli K12 genome, if you've got old 50 base Illumina data, you can't do any better than this 50 or 100 contigs, where each of these nodes is a contig, and the edges indicate the ambiguous relationships between them. As you increase to like Sanger size reads, you can get it into maybe 15 or 20 pieces, and the repeats that are now blocking you from finishing this genome are the big RDNA arrays and other large kind of segmental style duplications. But once you get above 7 kb for E. coli K12, you can collapse the assembly graph down into a single node, where that means I got one contig out of the assembler, and it's self-adjacent because K12 is a circular genome. It turns out by coincidence, 7 kb is also the magical threshold for human genome assembly because it's about the size of the human line elements. So if you can collect sequencing reads that are bigger than 7 kb, either for E. coli or human, you make the assembly process much, much easier. And so at the time, we were using PacBio data to illustrate these benefits. And it was expensive early on, so we started with microbial genomes and did a lot of analysis and demonstrated you could do complete assembly of microbial genomes. The next genome we moved on to was Drosophila melanogaster, which coincidentally was also the first genome that the Celera assembler was ever developed to run on. And this was showing the output of the assembly graph for our first melanogaster run using PacBio data. And it was as good of a result as we could hope for, where each of the chromosome arms was essentially in 1 or 2 contigs, with only some of the most complex regions like this histone cluster, which still remains unfinished in the genome to this day, unresolved. But each of these big colored squiggles is a single contig out of the assembler, which when you map back to the Drosophila reference, turns out it is correct. And it's also reconstructing more of the heterochromatic sequence, like the Y sequence. This assembly contained maybe 50% of the Y length, whereas prior it was only a couple of percent that was known in the reference genome. So there's a few problems that we had to address to do this kind of work. The Nanopore data and the PacBio data, by analogy, is very noisy compared to what we're used to. And so we developed strategies for correction and trimming of the data before you launch into the overlapping and assembly process. This is like a critical step to all assembly processes. And if you do assembly even with Illumina data, you know that read correction is really critical to getting a good quality assembly. I'm not going to talk about that step today. What we're going to talk about today is downstream from correction. Once you have the corrected long reads, how you go about overlapping them to generate these contigs. And the problem is the graphs that I showed you earlier are relatively manageable for E. coli, but when you get up to human, these graphs become very unmanageable. And you have thousands and thousands of reads with thousands and thousands of edges, and it can blow up the time complexity and the space complexity of the problem. So we needed to develop a solution to overlap these long, noisy reads, and we went back to this min-hash approach. The results on melanogaster were pretty striking. So the first assembly we ever did was using the version of CA we had at the time, which was version 8, and that took 630,000 CPU hours. So it ran on a cluster for basically a month to generate that answer, and we had a 15 megabase contig N50. And to show the speed improvement that we got from switching to the min-hash overlapping step, we got the same result in 500 CPU hours with actually a larger contig N50 of 21. So it's the perfect win in computation where it was orders of magnitude faster and it was a better result. And so it kind of demonstrates just thinking about these problems in new ways is necessary. So the latest version of the assembly software we've developed is called Canoe. It's a play on words because it's Celera Assembler, CA, new for long reads. Brian Wallens, who's in the top right, was a Celera Assembler developer at JCVI for many years. And when we hired him into my group, we tasked him with basically throwing away all of the old CA code that had been built up over the years for Illumina and 454 and Sanger and just restart the whole pipeline from scratch. And so in the end, he threw away 70% of the code base and rewrote only the bits that we needed to do this long read assembly. So it was a huge process that we started back around 2014 and just cut the first release last year. And the basic properties of the speed up is that we employ this lazy alignment process where we only use min-hashing approximation until the very end of the process when we need to compute an alignment, and you get a lot of time savings. So if we think back to the min-hash example I gave at the beginning, there's a couple of cool properties for overlapping that you can do. A, you only have to remember the sketches, you don't have to remember the sequences. B, you can actually weight the k-mer permutation. So if you see a unique k-mer, you can make the hash value give it a smaller value. If you see a repetitive k-mer, you can make the hash value give it a slightly higher value. And so this will weight your preference to prefer unique sequence away from repetitive sequence. And when you're doing hash table lookups, that's important to avoid these repeats that occur everywhere. And then you sample the k-mers from the union of these reads, and you can convert these samplings to both position and identity estimates to figure out the overlaps between two of these long reads. One of the observations that we made just last year was that if you use the min-hashing approach, because it's doing this random k-mer sampling, you can get away with using a relatively simple Poisson model for the survival rate of the k-mers. And so if this was my first sequencing read here in blue, and I apply some error rate process to it with a frequency d, k-mer size k, this is the expected number of k-mers that you would expect to survive that error process. With a little bit of algebra, you can turn that into a distance to recover what the error rate estimate was with this formula here, and that gives you a nice correlation identity estimate for an overlap, or if you're comparing two genomes to one another. So even though you're not computing an alignment, you can get a rough idea of what the identity is between those two sequences, which is really handy. And then once we need to, we can then compute an alignment on all of those to get the exact positions. We also employ a simple idea for overlapping. So the observation is when you have 50x coverage of long reads, the reconstruction process is relatively simple. You don't have to compute kind of and do a transitive reduction of all of the edges. If you just look at the best overlap for each of these reads, where best is the longest, you get a good reconstruction. And so as I'm showing here, the read at the top overlaps 3 other reads, but we really only care about this longest overlap. These other 2 thin overlaps aren't going to be important during the reconstruction because we're just using the best edges to reconstruct. So you can design your overlapping algorithm to be fast, but not necessarily sensitive, because you only care about these almost global alignments between 2 large objects. So it's easy to find those global similarities with min-hash, and then it's easy to build the graph with just this simple greedy algorithm to walk the best edges. There's a couple caveats that greedy approach, as all greedy approaches, can be misled. It's not optimal. And so up front, you have to very aggressively filter out overlaps that are false, meaning from one read to a different repeat copy. If you can remove those a priori, you can do a good job with just the greedy algorithm. And then you have to break at any unspanned repeats at the end to avoid any potential misassemblies. But if the reads are long enough, this is a nearly optimal approach. So there's a cool example here from Sergey Korn in my lab where he took the human microbiome metagenomic data set and ran it through Canoe with the overlap filtering turned off. So this is, you take all of the sequencing reads that came out of this metagenomic mixture, of which I think there's like 20 or so genomes, and you draw the sequencing, the assembly graph for all of the overlaps that you find. You end up with this kind of somewhat resolved but somewhat tangled mess where you've got this big component at the top where there's a bunch of genomes that have been intermingled and interassembled because of their shared repeat structure. But you use this as your first guess of what the sequencing error rate is of the process. So if you look at all of the best edges in this graph and just simply look at their distribution, you can see that most of them are less than 1% error rate between them. This long tail here is caused by the repeats and the false overlaps. So even just a simple filtering strategy that just says I'm only going to take overlaps that have less than, in this case, 1.5% error, and build my best overlap graph from that, you end up with this version, where now all of the things that were tangled together by the repeats have been separated out into separate components, and here each of the components is colored by the genome that they originated from, so you can see that each of the graph components is essentially a single genome that's been resolved out of this metagenomic mixture. This was our first pass. This is how Canoe currently operates. There's ways that we can make this much better and much more sensitive that we plan to work on. As I said, we can weight the min hashing approach, not only to avoid repeats, but you can also weight it to prefer haplotype-specific k-mers, so that repeats between a haplotype will be preferred over, overlaps between a haplotype will be preferred over overlaps between two different haplotypes. And you can also then compute actual alignments and look for site-specific variants to separate these out. And so MetaCanoe is next on the block. So what can we do with these long reads? Another flashback to the year 2000 when the Human Genome Project was announced here at the White House with Francis Collins and Craig Venter. At the time, it took international consortium and billions of dollars, hundreds of sequencing machines to generate the data. Celera had huge, alpha deck server farms doing the assembly process, and this was the result that they got out. So this is Human Reference 28, which was circa around 2000. Each of these horizontal lines is a contig, and we're changing the coloring anytime there's a boundary between two contigs. And so the fact that this is shuttered so heavily is indicating it's very fragmented. Places where you see down here, like on chromosome 20, there's a big arm of chromosome 20 that's into a single piece. All of the other stuff is very fragmented. This is how far we've come in 7 years. So Clive Brown, the CTO of Oxford Nanopore, sequenced his own genome on a couple of these MinION devices earlier this year. We ran that sequence through Canoe, and this is the result of the human genome we got for Clive. So this is 0.5 megabase per N50, so an average contig size of half a megabase, and 17 years later, 25 megabase contig N50 for Clive. So a personal genome. This is a diploid human. Presumably Clive is diploid. And there are chromosome arms that are in single contig. So this isn't even scaffolds. This is chromosome 2 here is all gray because the short arm of 2 is into a single contig from the telomere to the centromere. And so this is the power of these long reads with 55x of nanopore data. But it's not a completely solved problem yet. If you notice some of the white areas on this figure are the heterochromatic regions, as you can see here on 1, and then all of these short arms of the acrocentric, of which are 5, are all essentially just giant spans of ends in the human reference genome. And so being, the perfectionists that we are, we wanted to try and go in and resolve some of these. It turns out these are the RDNA arrays in the human genome. So in the 5 acrocentric arms, the short arm contains these tandemly repeated arrays of the RDNA unit, which is about 50 kb, and it's in 300 to 400 copies in each human genome and highly variable in terms of copy number. So it's a very difficult challenge to reassemble this. As you can imagine, it's, ribosomal RNA is very heavily conserved, and so these 50 kb tandem repeats are very difficult to assemble. But using both Nanopore and PacBio sequencing, we started to sequence actually some back clones recovered from these, and we're discovering there's a lot of structure and variation here that's currently gone undiscovered. And so if you know how to read dot plots, each of these green lines are indicating inversion structure. This indicates a deletion here of one of these repeat arrays. Here's a double inversion, another single inversion. So because you can imagine you have this big 50 kb tandem repeat array in maybe 50 copies per chromosome arm, there's a lot of opportunities for recombination and structural variation in here that we're currently looking into using these new technologies. And so this is the work of Alex Dilthey, who's been in my lab for the last year and a half. We've also started looking at more than just the clivome. So in a collaboration led by Matt Luce and Nick Lohman, we've also sequenced the GM12878 reference sample that's from this famous Utah-Ceph grouping. We didn't have quite as much coverage as Clive because we don't have free access to the reagents, but we still collected 35x of this reference genome, 11 kb N50 size, 3 megabase contig size. And I love this picture of Matt Luce sequencing DNA in the hotel room of AGBT a year and a half ago. And it was right after Nanopore released their rapid reagents. And so he did his library prep in the hotel room, and there's a step where you have to kill the transposase, and he used as a heat bath, he used the coffee water to kill the reaction and load this library onto a flow cell. So it really is a vision for portable sequencing in the future. And so all of this analysis, if you care about kind of the quality and what you're able to do with Nanopore data, this is up as a preprint on bioRxiv right now, Nanopore sequencing and assembly of a human genome with ultra-long reads. But we're still at 25 megabase and 50 length. That's not quite a closed human genome. So we were curious, how much coverage would we have to collect in Nanopore sequencing to get a closed human genome? So Sergey Korin ran an analysis of increasing coverage along kind of the x-axis and the N50 length of the expected contigs along the y-axis. And the clivome is up here with the CHM1 PacBio assembly, which is the best PacBio human assembly to date, and they're both about 25 megabase per contig N50. If we follow, just look at the blue line for this time. As you start to span longer and longer repeats, because you're collecting more and more coverage of these long reads, you start to climb this curve, and the contig size of the current human reference genome, GRCH38, is this horizontal line, just shy of 60 megabases. By Sergey's modeling, if we can collect 30x of these ultra-long reads that I showed you at the beginning from Josh Quick, our prediction is that we could get close to a reference quality assembly, at least in terms of continuity, just de novo from Nanopore sequencing data. And so we think there's a lot of gains to be had just by collecting more coverage of these very long reads. But still, you've got, even if you get 60 megabase contig N50, you're not guaranteeing that all of your chromosomes are into single pieces. And so we've been combining lately PacBio or Nanopore sequencing with Hi-C-based approaches to do this last level of scaffolding. We still can't get across the centromeres, for instance. And so when you get a bag of contigs out of the assembler, you want to know which two contigs came from chromosome 1. Using these Hi-C-based techniques, you can scaffold the chromosome arms together based on their proximal interactions. And so we demonstrated this on Papidum the goat last year with our colleagues at the USDA. And I think I can safely say we're the only ones that ever had a commissioned portrait of the animal that we sequenced. That was done by the daughter of one of the animal geneticists that we worked with. And we did PacBio, BioNano, Hi-C, and Illumina, combined all of those technologies together with our de novo PacBio canoe assembly, and generated essentially complete chromosomes for the goat at an average QV40 quality. And you can see the original contigs from PacBio, we had about 3,000. After adding those technologies, we're down to 680 contigs, and we're down to 31 scaffolds, because there was 31 chromosomes in this goat. So we're really starting to realize now de novo genomes where whole chromosomes are reconstructed into single scaffolds, which is really exciting to somebody who's been working on the assembly problem for their whole career. So to wrap up the assembly bit of this talk, I think haploid assembly, where you've got a single haplotype in your sample, is essentially a solved problem by long reads, because you just need to collect reads that are longer than the repeats that are in your genome, and it's easy to assemble. The greedy algorithm, if all of your reads are longer than the repeats, the greedy algorithm is optimal. And so just kind of with that in mind, I think it's the next conclusion that a complete human genome where you're spanning all of the centromeres and all of the heterochromatin, I think will follow shortly after the introduction of these ultra-long reads. So maybe 1 to 2 years before we close all the remaining gaps in the human, and I think it'll be done with things like Nanopore sequencing. And complete genomes will be the norm. So if you're a tool developer and you're working on sequence analysis, I'm starting to think about how we're going to adapt all of our tools to work on whole genomes rather than bags of reads, because you can get these de novo assemblies quite easily now. So the remaining challenges for me, I don't want to be out of a job now that assembly is kind of obsoleted. Luckily, it's still diploid genomes, polyploid genomes, metagenomes, populations like tumors and so on. These are still very difficult assembly problems that the long reads themselves don't solve. And so getting through the heterochromatin and large seg dupes in the human is still a challenge. And then finally, we had a couple of talks today on graphical representations of genomes. Once we have hundreds of complete genomes, how do we store them into graphical formats and allow exploration of the variation that we find in there? I think these are good problems to be working on these days. So I want to wrap up by talking a little bit about sequence alignment. So genome assembly and alignment have always kind of been my bread and butter and what I've been interested in. The overlapping problem that I just described, we're kind of doing global alignment between two reads. If we want to map reads to a genome, we kind of have to think about it as a local alignment problem. So I dug into my old deck of slides, and I found I had a slide from 2003 that was visualizing the process at the time. John was saying earlier, I just apply min-hash to everything. Before I applied min-hash to everything, I applied suffix trees to everything. And so when we were working on MUMMR back in 2003, this is how we did whole genome alignment. We would find maximal unique matches using a suffix tree between two whole genomes. We would cluster them, so you can see the gray guy here is not like the others. The other ones are in a consistent ordering, so we use a clustering algorithm to toss that one away. And then we simply use a banded dynamic programming algorithm to collect those two clusters and extend off of the ends to make high-scoring local alignments. And this technique has been around for ages and is still a great way to align two whole genomes, and you get fairly accurate results out the end. The problem is that suffix trees, suffix arrays, Burroughs-Wheeler, these type of techniques are really designed to align highly similar things. So if you're like 99% similar to what you're aligning to, you have very large mums, and you save a lot of time by getting those exact matches out of the suffix tree and not having to do extension, via dynamic programming on everything. And so we start looking at Nanopore data. These approaches get relatively slow. Blazor, for instance, uses suffix array data structure for doing its seeding and extension. And if you give it Nanopore reads, you have to wait a while to get results out the other end. And so if we're getting data off of this Nanopore device in real time, we can't wait around forever for the algorithms to align it. We want to know the answer as soon as the read came off of the Nanopore. And so I had a summer student last year who's a grad student at Georgia Tech, Chirag Jain, in the top left, and paired him with Alex Dilthey, who's the postdoc in the lab. And last summer they worked on this problem. If you get a stream of reads off of the Nanopore, how can you quickly answer questions like, what species did this read come from? Is it a known or a novel species? If I map all of my reads to the genome, do I see uniform coverage, or do they all pile up in one region? And I want to do this in real time, and I want to do it fast enough that I can interface with the instrument if necessary, and I want to know if my database that I'm mapping to is adequate. And so we have two choices. You've got the full alignment-based methods like NUCMR, which I just demonstrated on the previous slide, which does the seed and extend method, and tools like BWA-MEM operate very similar to my NUCMR tool. On the other end of the spectrum, we have the signature-based metagenomic approaches that just look at bags of k-mers to determine where a read or a sample came from. But you're slow on the one end, you're fast on the other end, you're accurate on the one end, you're inaccurate on the other end. So we were trying to kind of split the difference and use these minimizer and min-hash based ideas to have an approximate alignment tool that could map reads very rapidly to a reference database, give you both a positional and an identity estimate, but still be fast as some of the k-mer-based approaches. And so Chirag formulated this problem as, if I have a sequencing read that I want to map to my reference genome, we want to be complete and kind of know all of the places it could possibly go. So given a read, I want to return all reference windows of the size of the read that have an identity greater than some minimum threshold, which you provide as a parameter. So for Nanopore data or PacBio, say 0.85 is our minimum identity parameter. And Chirag's algorithm will return to you all of the places your read can go with that identity, modulo, that it's an approximation algorithm, so you won't get every single one, but you'll do pretty well. And it's hundreds of times, in some cases 300 times faster than BWA-MEM mapping for, say, PacBio data to human. You can support large reference databases. It auto-parameterizes itself, so you just tell it the length of your reads and you tell it the identity of your reads, and it can auto-configure the algorithm to find and be sensitive enough for that. It reports kind of straightforward ideas of mapping quality and so on. With the limitation that the results that you get are just a position and an identity estimate for the read, not the actual pairwise alignment. But if you know the position and the identity, you can then call Smith-Waterman or something on that region to get the gapped alignment if you want it. In the first version of this that Chirag wrote last year and that I'm presenting now didn't implement split mapping or local alignment. It's really globally aligning a read to a substring in the reference sequence. But split mapping is in progress. So here's how it works. It's kind of a combination of both this minimizer idea from Roberts and York and the min-hash idea from Broder. Also, a comment on the minimizer. I think we were the first ones to realize that the minimizer and min-hash were fundamentally kind of the same idea, and it took 10 years for anybody in the field to notice this. If anybody noticed it before us, please let me know, but nobody published those same two words in an article prior to 2014. And prior to the York paper, there was also another technique called winnowing, which was taking min-hash and looking at only a single value and decomposing it into a set of minimizers, essentially. So this is evidence to me that this is a brilliant idea because people keep falling over it in different fields. But when you look at the math, they really are equivalent. And so you might hear me call winnowing and minimizing interchangeably. So you take your reference genome, you scan a sliding window across it, and you collect all of the minimizers, and this forms your index. And so you can see where the space savings come from, because you don't need to remember every Kmer in every position, you're only remembering the minimizers, and this gives you some fold reduction in what you have to store. And you keep that both as a hash table, so you can look up by Kmer, and you store those minimizers in a sorted position list. Then given a read, you run the same process on it, collect its minimizers, look up its minimizer matches to the reference in a hash table, and you get those matches back as a sorted list. So all of the minimizers that the read shares versus the reference is a sorted list. You just have to scan through that sorted list and look for any set of minimizers that are close enough in physical space in that list that it's higher than you would expect by chance. And so in this illustration here, we have 2 minimizers that are separated by 1 Kmer, and this is triggering a more exhaustive search. And so it's a very fast, just linear scan of this match list to determine the areas that have significant hits. So that gives you your candidate mapping regions. Then we go back to those regions and we do a sketch of all of the minimizers that are in there to compute the identity like I described for the overlapping process. So in this case, I have 2 minimizers that are shared between the reference and the read, and there's 3 other minimizers that aren't shared. And so as I described for the min hashing, you take the union of your minimizers, and you take the intersection of the minimizers, and you look at that ratio, run it through the statistical Poisson model for the Kmer survival rate, and you can get an estimate of identity. And so for each read, you look up its Kmers, you find this positional scan, you compute the identity, and you immediately return the bounds of that best hit and the identity of that best hit. And figuring out the window size and the Kmer size, all of this gets auto-parameterized based on the math modeling. So Chirag actually presented this at Recom, so this might be familiar to some of you if you were in Hong Kong this year, and it's out in the Recom proceedings as a fast and approximate algorithm for mapping long reads to large reference databases. And so we wanted to do the experiment to map long reads to large reference databases. And so we took the Human Microbiome Project data again, so this is PacBio data from real DNA, it's just a synthetically mixed community. And we mapped it against essentially all of RefSeq with MashMap. To compile the truth, we took the known 21 species that were in that synthetic community, and we used Blazor to map all of the reads just to those 21 species. But MashMap didn't have that advantage. We let it map all of the reads to all of RefSeq. So we're talking about a 20 gig database versus a 60 meg database. And MashMap took 3 CPU hours to map against all of RefSeq versus Blazor taking 80 CPU hours to map to only the 21 genomes that are known truth, right? To make it even harder, there was 3 genomes in that HMP sample that weren't in the RefSeq database, so we were handicapped even a little further. Here's the output of an abundance estimation from that mapping, just to show that it's doing essentially the right thing. So the dark blue are the MashMap estimates of abundance based on the number of reads that were assigned to the different species. And this is measured at the genus level. And the light blue is the truth mapping against the known 2021 HMP samples. And so you can see pretty much in agreement for all of them, this little blue nub down here are the reads that MashMap align to things that aren't in the reference, that weren't in the synthetic community. So this could be contamination or it could be false positives. But this number 2 here lights up as being the oddball. And so it turns out this is Actinomyces, and it's one of the genomes that did not have a near neighbor in the RefSeq database at the time. So essentially, MashMap doesn't have anything good to map it to because we didn't know of this genome in advance. And this is a huge challenge in metagenomics, right? This unknown genome problem. If I do a metagenomic analysis and there's genomes in my sample that I've never seen before, how do I know that? And the Kmer-based approaches are often confused by this and will make incorrect predictions of what is in your sample because they've never seen these Kmers before, so they don't know what to do. The really cool thing about doing a mapping-based approach, so if you can afford to do this quick mapping, which now MashMap makes feasible, you get the identity and the positional estimates. And so if you dig in to the histograms for the identities of these mappings for Actinomyces and Neisseria, for instance, on the right-hand side is Neisseria. We know this genome was in the sample, okay? I'm showing identity histogram on the X here, number of reads on the Y. And this is reflecting essentially the read error, because these were PacBio reads. We were mapping them to the actual truth reference. And we see that, you know, you have a mode of maybe 90% identity with this tail. This is just what PacBio error rate looks like. And so Neisseria is something that's in there. If we know that's what the histogram looks like for something we know, then we can look at Actinomyces histogram and say, this looks bizarre, right? This doesn't look what I expect the PacBio error rate to look at. So now I can build a model that predicts this as a novel genome because from the identity alone, I can tell that it's not like the thing that's in my database. And so Alex is currently working on the modeling for this using both the identity and positional estimates to determine if my mappings to my reference database are true or not. So what's the probability I sampled this read from that genome if that genome's actually in the database? So I think this is kind of a powerful approach to do metagenomics with long reads, because you can distinguish these novel things from the known things. And then to wrap up with MashMap 2, Chirag came back again this summer, and so the next logical step was to extend this kind of global alignment problem to the local alignment problem. And so you think about how we constructed it. We took a window, which was the length of the read. So to do the local alignment problem, we're just taking a window that's some 10 KB or 5 KB substring of the read, slide that window across the read and find all of the hits for those windows. So it's kind of a rough approximation of local alignment. And you can then use this for split read mapping, or you can even use it to do genome to genome alignment. So I just asked Chirag this morning for this example where you take a human reference genome along the x-axis, and you take all of your assembly contigs for your de novo human genome and you want to map it to the reference genome. So with his MashMap 2, which we should be releasing within the next few months, running on 8 threads, the indexing takes 30 seconds, the mapping takes 50 seconds for this assembly. And so you've got this dot plot in just over a minute of wall clock time. And so for people that do assembly, we make so many dot plots because we're always trying to validate if our assembly is correct or not. Being able to generate these for human scale genomes in a minute is super handy. So where are we going? So the alignment summary, long sequence alignment, which I started with in my career, is in vogue again. So it's good news for me. We can go back and apply these same methods that we were using 15 years ago to align these Nanopore and PacBio reads. As I mentioned earlier, the suffixes were good when things were similar, but when you have divergent sequence or noisy sequence, we find that hashing works much better. In particular, this kind of locality sensitive hashing strategy where you take like a 10 KB piece of sequence and you reduce it down to some compressed representation like we've done with min hashing or minimizers, allows you to rapidly scale up the mapping problem. And so Hang Li has also worked along similar lines. So he has a tool called Minimap, and Minimap 2 was just released last week, which uses a minimizer-based index and clustering on those minimizers, so very similar to these ideas. And we use the minimizer plus min hash combination that I mentioned here, which gives us uniquely the ability to predict the identity for the positional estimates that we're returning, which Minimap doesn't currently do. Recently, Hang has added to Minimap 2, once you return these estimated regions of homology, he'll then perform a very rapid pairwise alignment. So if you want to do these kind of rapid dot plots and get rapid sequence alignments, Minimap 2 is a good option. So the remaining challenges, trying to do this in real time for mapping and metagenomics, we're working on the modeling to predict abundances accurately and to predict what reads are coming from unknown and potentially novel organisms. To apply this to obviously doing lightweight and accurate SV detection in human genomes and so on. And then finally, graph mapping and multiple genome alignment. Just like metagenomics is the next frontier for genome assembly, dealing with multiple genomes in the alignment space is still a challenging problem. So if we've got, say, 1,000 vertebrate genomes, can we efficiently align them to one another and generate a graph representation that we can use for analysis? So what's next? I'll just end with a couple of forward-looking statements. So first of all, I like this picture on the right. So the cover that I showed you earlier for the genome assembly issue was developed by the Cbased team from St. Petersburg. This was the runner-up for that issue, which was genome as needlepoint. So Jason Chin had an assembly graph for one of the human genomes he had generated with his Falcon assembler. That went to the team at EBI, and they have a graphics designer there, Spencer Phillips, who put it into needlepoint and has this home sweet genome picture that I like, and it fits perfectly. I'm at the Genome Institute. This hangs on my door outside of my office. This is what the human genome graph looks like with PacBio data. It's still pretty tangly. So can we use Nanopore data to have a finished genome where we get through all of the centromeres, all of the RDNA arrays, all of the heterochromatic sequence, and finally finish all of these gaps off? And I think that's feasible now with these ultra-long preps. And so we're working hard on trying to do that, and I think we'll get there in the next few years. If we have whole genomes for all humans, all microbes, people are carrying nanopores around on the street and sequencing their bathrooms, how do we search and compare the billions and billions of genomes that we'll have as a result of that. And that's where I like these kind of min-hash style approaches, because they scale wonderfully. What are the killer apps of portable ubiquitous sequencing? And I don't have the answer for this. I mean, just the fundamental change in how you think about sequencing, where it used to be you'd send your sample to the core, they would sequence it and send you the result back, to now you can just kind of do it. They've got devices you can plug into your phone and do sequencing as you're walking around in the field. What are the new applications that are going to be opening up completely new problems that we haven't thought of before? And then the problem that I think a lot about these days is this MinION device costs $1,000, right? I think a lot of people in this room have $1,000 they could blow on a sequencer and start doing sequencing. I'm a computer scientist. I've never bought a piece of lab kit. The Nanopores are the only piece of lab kit I've ever bought. And I don't say that as an endorsement, because I'm a federal employee. I can't endorse anything. But to think about that, if you have all of the undergraduate classrooms, all of the high school classrooms in the world, you've got all of the scientists doing sequencing essentially in their kitchen, how do we collect all of that data and centralize it in a way that we can build knowledge from it? If all of us go out and sequence tomorrow and we find novel genomes and novel discoveries and we don't share it with one another, we get no benefit from it. And so how can we incentivize the sharing and avoid the siloing that's going to happen now that the sequencing itself is democratized? And so we have a couple of ideas, but I think this is a problem for all of us to attack and is really kind of an open data problem that we need to think hard about. I'll say I think the one key is we need to reduce the barriers to entry as low as possible. So if there's a way we can make it as easy as possible for people to have data shared and incentivize them to do that, those are the keys. The current model of, you know, the GenBank submission system probably isn't going to cut it. So I'd just like to acknowledge my great team at NHGRI, who I'm super blessed to work with, and primarily Sergey Korin and Brian Andov, who are here with me this week. They have posters, so go talk to them. Brian did a lot of the MASH work and also developed this Corona tool that you might be familiar with for metagenomic visualization. His poster is essentially now, poster B22. And Sergey has a poster on all of our applications of min-hashing, B050, which I think is tomorrow. Brian Wallens, who does all of the canoe development, Alex Dilfy and Arang Ri, who are the two postdocs in the lab working on assembly and haplotyping problems, and Chirag Jain and Anna Sappington, who have been my wonderful first ever crop of summer students that have done fabulous things. And Alex will soon be leaving us and returning to his home country of Germany, so your name here. We would love to work with people that are interested in solving these types of problems and answering them with these kind of computational approaches. So if you're interested at all working at the NIH, we have a great visiting fellowship program. Please just contact me here or send me an email with your interest. And many of our collaborators that provide all of this data for us. And with that, just thanks so much for having me here, and I'm happy to take any of your questions.
Thank you so much for this great talk. You're welcome. I have many questions, but probably we can let the audience first to formulate theirs. Yes?
Yes, hello?
Yes?
Okay. I was wondering, MeshMap was designed for genomes. Did you try using it for transcriptome, long-read transcriptome data?
It's a good question. We have not done that yet. There's the suite of related tools like Callisto and Salmon and so on that use equivalence classes to determine where transcripts are coming from. I think a lot of those would work pretty well for long reads as well. If you want to have this mapping of transcriptome data to genomes, I think it could be useful for finding novel splice forms and things like that. The one caveat I'll say is that MashMap is designed to find very large chunks of sequence, and so it's optimized for like 5 or 10 KB window sizes. And so it's probably not well designed to find small exons in larger sequence. I think that's a harder problem that would require more compute time.
Are there any insurmountable limits to the maximum read size you think you'll be able to get to the nanopore, or is it feasible that we could one day sequence chromosomes with it?
Yeah, I think that's the vision. I mean, currently it's handled with care. I mean, it seems that the nanopore itself is capable of sequencing whatever you can get started in it. And so really it's sample handling, like how you can extract giant molecules and how you can get them onto the pore array without fragmenting them. So just simply pipetting and things like this causes fragmentation. And so there's a lot of kind of hobbyist R&D going on with the user community now to find the longest molecules. And 1 megabase is the top end right now that anybody's seen, but I think we'll see higher.
It was great talk, thanks. I wonder, there is a lot of gaps in current genomes which are not because of the repetitions, but because those regions are not readable by current techniques. Do you think that the Nanopore really solved that? Like G4, for example, did you try any bare genome which are heavily fragmented?
Yeah, so we haven't noticed any obvious biases towards what it is and is not able to sequence. There might be, weird secondary structures that could affect the sequencing ability of the pore, but just as far as simple things like GC content, we've been able to get through low complexity and GC-rich areas without too much problem. So I think it's a big improvement over what's currently available.
I have a question about metagenomics from the long reads, and then in particular looking at this error profile to distinguish novel from known organisms. What about the semi-novel organisms, like divergent strains of viruses, where this divergence is quite high?
Yeah, so I think the observation is if two things are very different, it's very easy to see that they're different, but if you're trying to tease out novel strains from a mixture, this is a little harder. So I think what you could envision is using one of these approximate tools to essentially recruit a set of very similar genomes, and if you care about strain level diversity, once you've identified that I have a genome in my mixture that's very similar to, strain A, then you can go into these mappings and compute the pairwise alignments and try and pull out SNPs, and then you want to resort to phylogenies to reconstruct their relationship to the known database.
Thank you.
Well, I have actually a question related to this one, because the example that you showed, it was kind of a toy example. So you have a simple mixture of genomes, right? And then one of them was unknown to your database. But I mean, the regular situation in an environmental sample, for example, is that you will have many of unknown genomes there. So maybe most of them are unknown, okay? So how does it work in this case? So how are you going to detect how many different microbes you have in your sample that are not known, and how you find them back, and I mean, how you go in that scenario?
Yeah, thank you for keeping me honest, not letting me get away with 20 genome mixtures. Absolutely true. So the first observation is that there is a lower limit to MASH map sensitivity, and so we ran this with 85% identity. So if there's novel things in there, particularly viruses, which mutate very rapidly, you might not actually align them to anything. And so you could have these unassigned genomes. The other observation is if you do have alignments, you can compute essentially quality values from the mappings. And so because it's this min-hash process, you can ask the question, if this read actually came from this genome, what's the probability that I would have seen this many minimizer matches? And then you can use that as a quality value. You can normalize across all of the mappings of that read to your reference database, and you can use that to figure out where it likely came from and inform kind of a phylogenetic placement just based on the identity. That would let you make a prediction, if you build a model that knows how to look at that, make a prediction about if you have a new thing and what genus it's potentially in, but you wouldn't be able to count it with that model, like how many new species there were in the sample. And so it's not necessarily trying to address that. And I think it is an open problem that this isn't necessarily solving, but I think it is the most important problem in metagenomics right now, because metagenomics is all about the database. All of the methods, if they had a complete database, if we knew everything that was in the world, metagenomics would be an easy problem. And so how we can develop models that are aware of the limitations of the database is a problem that we're thinking about, but we won't solve it alone.
More questions from the audience? No? Okay, so then let's thank Adam and the speakers
Thank you.
for today again.